Hello, this is Anuja Matthew. It's October 2nd, 2020. Today I'd like to talk about monoclonal antibody therapies. This is a form of immunotherapy that uses monoclonal antibodies, which is a single antibody that's able to bind either cells or proteins or viruses. These monoclonal antibodies are injected in large quantities into humans with the idea that they might either reduce inflammation, treat cancer, or block viral infections and decrease viral loads in case of infectious diseases. There are many monoclonal antibodies that are being used on patients currently. These are just examples of three monoclonal antibodies. Humira, which targets a cytokine that's used to treat arthritis and arthritis-like conditions and even Crohn's disease or ulcerative colitis. Dupixent, which also is used against a receptor for a cytokine. It's used to treat atopic dermatitis and asthma. And Keytruda, that targets a molecule known as PD-1 that's used to treat many kinds of cancers. So these are just examples of successful monoclonal antibody therapies in the market already. This is a figure showing a list of viral and bacterial pathogens that can cause pandemics or epidemics, and looking at the number of monoclonal antibodies either at the preclinical stage or different phases of clinical trials. Most of these diseases have probably one or two monoclonal antibodies that are currently being tested at the preclinical stage, shown as these light green bars. So for MERS, SARS, influenza, Zika virus, and now COVID, there's over 10 or 15 monoclonal antibodies that are at the preclinical stage that are being tested. And I'd like to talk much more in detail about monoclonal antibodies against SARS coronavirus 2. How does one make a monoclonal antibody? For COVID-19, typically you take blood from a person who's recovered from the disease. You use a bait or molecules to fish out B cells, which are the antibody secreting cells, and they produce antibodies. And you're specifically looking for key portions of the novel coronavirus. In this case, it's the spike protein, and particularly the receptor binding domain that will block infection. So then you decipher the code for the antibody. So you do extensive genetic sequencing, and then you introduce this DNA into cells, and you can mass produce antibodies. So many researchers produce hundreds of monoclonal antibodies and then test them effectively in the lab using very sophisticated analysis to try to figure out what the best antibodies are and whether they can actually neutralize or prevent infection. So in the case of COVID-19, the spike protein on the surface of the virus binds the ACE2 receptor present on human cells. So the idea is if to neutralize infection, if you have enough of neutralizing antibodies, in this case a monoclonal antibody, if that neutralizing antibody can bind or block the spike protein at the right site, then it'll prevent the virus from binding the receptor and therefore prevent infection. One of the companies that's actively involved in generating monoclonal antibodies for therapy against SARS coronavirus 2 is Regeneron. If you need details about the antibodies, please go ahead and look at this article that was published in Science a month or so ago. But Regeneron has generated a combination of two monoclonal antibodies that are designed specifically to block the infectivity of SARS coronavirus 2. They've then batch produced very large quantities of these monoclonal antibodies that can be used for therapy in humans. It's a very expensive process, and typically antibody therapy can range from $15,000 a year and in some cases for cancer immunotherapy up to $150,000 a year for treatment. Regeneron posted the results of a phase 1-2 trial on non-hospitalized patients on their website. As far as I know, this information has not been published yet in a peer-reviewed journal. But about 275 people were enrolled in the trial. They were randomized to either to receive a high dose of the antibody cocktail, a low dose of the antibody cocktail, or placebo. People in the trial were of mixed ethnic origin. More than 50% had underlying risk factors for severe COVID-19. The mean age was about 44 years of age, and there was about an equal participation of male and females in the trial. So in this trial, basically, people who had mild symptoms, who had viral loads that were detected by nasopharyngeal swabs, were enrolled in the study. They measured viral loads. They looked at serological status and then compared to see, does the antibody have any effect in reducing the amount of virus and using these swabs. So the serological status highly correlated with baseline viral load. So what does that mean? So there are some people who were able to generate their own immune response as is expected. And in those people, they were able to control the virus without much effect of the monoclonal antibody therapy. And that's expected because if you generate a good immune response, you should be able to clear a virus. And we are assuming in most people who get the infection who have mild symptoms, that's what's happening. Serological status at baseline also predicted how rapidly patients had alleviation from their COVID symptoms. So there was a group of people who did not have antibodies directed before they were given this monoclonal antibody therapy. So they were not able to generate a strong immune response by themselves. In these people, the monoclonal antibody actually had an impact. So because they didn't have their own antibodies, when given this powerful monoclonal antibody cocktail, they were able to reduce viral load significantly. And I'm not spending too much time going into the data because this hasn't been published in a peer-reviewed journal. They've provided some information on their website, but what they say is patients who were seronegative, that means patients who did not have their own immune system generate antibodies at the beginning, or who had high baseline viral load. So if they had a high titer to begin with, those people had greatest benefits in terms of the antibody. So the cocktail was beneficial. It reduced symptoms in individuals. It reduced viral loads in individuals. So this is very positive as this monoclonal antibody seems to be effective in reducing both symptoms and viral loads. There are two companies that are currently in phase 3 trials that are enrolling patients to determine whether experimental monoclonal antibodies can prevent infection from SARS coronavirus 2, and they're enrolling healthy adults. So one is the company that I just talked about, Regeneron, together with the NIH. They are trying to determine in a larger number of adults, so over 2,000 asymptomatic adults, and these are household contacts of people who have active SARS coronavirus 2 infection, to see whether the monoclonal antibodies can actually prevent infection. They're also going to be doing trials in hospitalized patients. So the first trial was done in non-hospitalized patients, but they're also trying to see whether monoclonal antibodies can be effective in hospitalized patients who tend to have higher viral loads. Another company, Eli Lilly, together with the NIH, is also looking at a specific monoclonal antibody that they isolated and characterized. And here again, there's about 2,400 participants that will be given either this monoclonal antibody or a placebo to see whether it's effective. And as far as I know, this trial might be targeted at patients who might be at nursing homes as well as patients who are hospitalized. So they are looking at a larger number of patients for the Eli Lilly trial. So there are many monoclonal antibodies that are being currently tested at the preclinical stage. A number of companies will get into the market. Right now, Regeneron and Eli Lilly seem to be ahead, but AstraZeneca is also preparing to do trials. So it's a costly way to generate therapies. It might be effective. We still have to wait and see its early stages, but this is one way of trying to prevent infection or block infection if until we get an effective vaccine, which might be early next year. I would like to end with these sobering statistics. There's been over 34 million cases to date and over 1 million deaths. It will not be possible to treat people who are sick worldwide with monoclonal antibody therapies. This is a test and will be used, if effective, in a select subset of individuals. Thank you again for your attention, and I hope you've learned something new today.